Hello and welcome. This presentation is part of the Myothermus ruber Genome Analysis Project under Dr. Lori Scott at Augustana College in Illinois. Today we are going to be comparing a set of genes found in Myothermus ruber and Escherichia coli, specifically mRub 680, 836, and 837, compared with the E. coli genes for CCMA, B, and C. These genes are involved with the biosynthesis of a protein called cytochrome C. Cytochromes are a large group of proteins in bacteria that are responsible for the transport of electrons between proteins. There are a large variety of different cytochromes, but today we will look into C-type cytochromes, which are defined by the assistance of a heme cofactor in their mechanism. The biosynthesis of C-type cytochromes requires that a heme cofactor be exported from the cytoplasm as the formation of the heme and cytochrome C complex occurs in the periplasm. The genes we are concerned with today are part of an ABC, or ATP binding cassette, transporter system. In the diagram, this system is labeled with the letters A through D, as seen here, and was originally thought to export heme from the cytoplasm into the periplasm, where the cytochrome C biosynthesis occurs. For this system, CCMB and CCMC form the intermembrane exporter, while CCMA acts to transport heme to the CCME, a chaperone protein, which is involved in the further steps of the cytochrome C biosynthesis mechanism. CCMD, then, is a helper protein, assisting CCMA in the heme transfer when it is present. Now, although we know that CCMA and CCMD interact with the heme cofactor, research has shown that the BC complex does not actually export heme and that its substrate is unknown. As I mentioned, we will be examining these genes in Myothermus ruber to see if they compare to the known proteins in E. coli. Now, M. ruber is a red pigmented gram-negative bacterium that is closely related to the genus Thermus. It has been known to form colored biofilms on machinery and lives in thermal environments. It is important to study its relationship to E. coli in hopes of determining some additional evolutionary relationship and expand on our knowledge of its genome. In order to compare these sets of genes, a large selection of bioinformatics tools was used. This included tools for determining gene sequences, identifying alternate start codons, cell localization techniques, locating functional domains and families, confirming the presence of operons, and comparing orthologs and paralogs between and within genomes. These tools are provided to the public online for free use for any who want to learn more about genomics. Looking at the protein CCMA, it was determined that mRub 680 and E. coli gene B2201 are orthologs that code for this gene. This was determined using BLAST-P, which compared the sequence of E. coli B2201 to the M. ruber genome, and in such matter isolated mRub 680 as the top hit. This protein, CCMA, is found in the cytoplasm attached to the inner membrane, interacting with the BC complex. As seen in the table on the right, the data confirms that CCMA in both species contains an ATP binding domain and is involved in the transport of the heme cofactor. This is clearly seen in the tiger fam match, right here, which indicates both ATP binding and heme involvement. It is important to note that the expect values, or E values, are very low in all cases. This indicates a high match and a high significance of the data collected. One interesting match is the 1Z47PDB match, located at the bottom of the table here. This match is for the ATPase subunit of a component of another ABC transporter system, cis-A. These ATP binding domains are part of what characterizes ABC transporters. The ABC transporters are driven by ATP hydrolysis, and so there must be a component that can accomplish this. In the cytochrome C biosynthesis pathway, that is done through the ATP binding domain of CCMA. Now the results for CCMB are a little different. As I mentioned before, research has confirmed that heme is not the correct substrate for the BC complex, and the real substrate is yet to be discovered. However, this finding is rather recent, and so many of the databases used still list CCMB and CCMC as heme transporters. We know that they are involved in the heme system because of the interaction with CCMA, which binds heme, but the exact mechanism is still undetermined. Nonetheless, the BLAST-P confirmed that B2200 and MRUB836 are orthologs that code for CCMB. Being a transporter, CCMB is located in the cytoplasmic membrane and also is known to contain 6 transmembrane helices. The data displayed to the right shows the association between CCMB and the heme exporter, which has since been disproved, as seen by the diagram definition. This is still strong evidence, though, that B2200 and MRUB836 are orthologs since they have all the same top hits from the databases, each with extremely low E-values.